everybody. Welcome to the second podcast in our regulation domain. In this one, we are going to break it up into two parts. This first part is going to look at development from an animal perspective. Basically how this one came around. So here you go. Let's get it rolling. Okay, there we are, developing from an itty-bitty single cell to a baby. So here are the big questions we need to look at and answer. How we get multicellular organisms from a single zygote, how development of an animal is different from plants, how they're the same, how parts are positioned properly in the organism, and how differentiation of cell types occur in both animal and plants. And then how does genetics play a role? So this is going to bring in some of the ideas that we learned in the last one and the effects that the environment also has. So strap in, here we go. So let's look at animals first and kind of give ourselves an overview on what happens. All right, so we know we've got the initial first cell that starts. Okay, so we have the initial first cell that begins. It occurs in various stages, and development doesn't end once you're born. So development is something that is different from growth. So how do we compare development to growth? Growth is change in size. Development is actual change in structure and in function. So we know that we go from the gametes that fuse together on fertilization. That single zygote then cleaves multiple times into becoming a ball of cells. You get gastrulation, which is where you begin to form the blastula, and then you have the different formation of the embryo, organ formation, ultimately the production of the kiddo. Okay, so external development. Okay, you're going to have things like frog eggs. They develop externally from. Okay, so you've got the frogs that are going to develop externally from the parent. So you've got the little embryos. You can see the yolk that's providing energy, and then you have the developing little kiddo. Okay, internal development. Okay, you have things like here would be what's going to become a little puppy dog. Okay, so you have the embryo, and then you have the developing placenta in the umbilical cord. Okay, so the placenta is providing, it's basically the connection piece between mom and baby, and that's what's going to help diffuse across nutrients and so on. All right, so the idea of you have, not only just the embryo that plays a part in development, but you also have a number of other structures that help supply energy. So you have an amniotic egg. This evolution was hugely important for things like reptiles, birds, and mammals, and so on, to spend more of a terrestrial life versus having to form, because that amnio is going to maintain moisture for the embryo, where it means easy when you're a fish, right? You're surrounded by water. So you've got the four areas of the membrane: the amnion, the chorion, the allantois, and the yolk. Okay, so the amnion, that's going to be that protection, sort of shock absorption, moisturizing fluid that's going to protect the fetus. The chorion is going to be important for gas exchange. So remember, we've got to get oxygen in, CO2 out. The allantois is going to look for waste storage, so any kind of nitrogen waste, stuff like that goes in there. And then the yolk serves as an energy source, because we know this kind of stuff takes boatloads of energy. So in the beginning, there's fertilization, right? So the fertilization piece.
Nothing but rocks.
So in the beginning, there's fertilization, and you've got the male gamete, female gamete coming together to form your zygote, right? So in sea urchins, it's a little something like this. Okay, you get releasing enzymes from the male gamete that's going to break through the egg and basically allow for that combination of dad's chromosomes and mom's chromosomes. So the fusion of the sperm and the ovum are going to trigger the formation of this sort of fertilization envelope. Okay, that's going to happen. And this creates a block so that you don't get other sperm fertilizing the same egg, because that would cause mega problems. In animals, it's kind of similar. There is a protection of cells called, basically all these follicle cells and these layers that exist around a mammal egg, and the sperm has to like dig its way through using enzymes and so on that are going to push its way through for that. So take a second, go back and look at the other one and see if you notice any differences that occur. When you have the fusion of these gametes, you're going to trigger massive calcium ion release across the ovum. This is going to help to protect that newly formed zygote from any other sperm from getting in, okay? Because what we don't want is polyspermy, and that's going to be multiple sperm, so then you're going to get a wacko chromosome count that's going to that's going to happen. Okay, so after fertilization, you can see the formation of this occurring not too long. Okay, and that's a slow block. Okay, and that's the results you get. Okay, so taking a look at how this calcium ion release, okay, basically blocks any other sperm from coming in. Okay, so there's our zygote. There's your throwback Thursday right there, babe. All right, so all the blue light micrographs depict sea urchin embryos, okay? And just so you know, so the blue ones are sea urchins, okay? So that's single cell. Then we get cleavage. Cleavage that happens is the division of the cells. These are the first mitotic divisions that are going to happen. The zygote's going to divide from 2 cells to 4, dividing again, and so on and so on and so on, where it becomes this ball of cells. Okay, and again and again and again. But note something really, really important here. Notice volume isn't changing. Cell size is changing. Volume isn't changing. And at this point, each cell can become any organism. This is what we call totipotency. Okay, any of these cells I could break away and it would divide and become that organism. Hence where we get the idea of identical twins. Okay, so here's a little cleavage action. Go ahead back and you can watch it in the Prezi. Okay, and eventually you go from being that zygote into the ball of cells, which is your morula, and then you get streaming of those cells. They're going to stream outward, creating a hollow ball, and that formation from that hollow ball, morula, right, is going to form the blastula, which is going to have an inner mass of cells, and that is what's going to become the actual embryo itself. Moving from that into gastrulation. Gastrulation is where we have now an organization of the cells. This process is really important because you're going to form the primary germ layers that are going to be established in order to create the right areas of us, okay? And all tissues and organs derive from these initial germ layers, okay? And that's how basically we all form. So this is a kind of an important piece, and a lot of critters go through a similar process. Okay, so in a sea urchin, here you have sort of the structure of what goes on. You have what's going to become the ultimate critter, right? And you can see how they're forming and how they're going to develop from that initial set of cells. And the color indicates the different germ layers, okay, that are going to become the actual organism. So take a second at like that. In frogs, you can see that you have areas that are already beginning to differentiate what will become differing areas. You've got an ectoderm, a mesoderm, and an endoderm. Those are generally the 3 germ layers that are pretty consistent among all living things. Ecto, outer, meso, middle, endoderm, inner. So, early gastrulation, this early formation of these germ layers, you get a mid-gastrulation, complete gastrulation, which is the complete formation of all those germ layers that are then going to become your critter. And then you get a little urchin. Isn't it cute? So here it is, happening in realness. So go back and take a peek. So the fates of the 3 germ layers, okay, you need to understand this, and you're going to need to know this. So take a second and, stop here if you have to, go back and write it down. In the ectoderm is the outer layer. This is where you're going to get skin formation, sweat glands, hairs, your nerve and sensory systems form from this, teeth, some of the early, some of the germ cells that are going to go on to become other parts. Your pituitary gland and adrenal medulla. Your adrenal medulla is sort of the outer part of your kidney. Your adrenal gland part that sits above your kidney. So the pituitary gland, that is an important gland. All right, picture this. You find your pituitary gland. It dangles down from your brain. So picture if I could go from between my eyes here, find your occipital protuberance back behind your head, and you go back in. So if I were to kind of, as weird as this sounds, if I were going to go bore right through it, where my two fingers meet, that is the general location of your pituitary gland that basically dangles down from your hypothalamus of your brain. Now your mesoderm, which is the middle layer of the embryo, this is going to become your muscle and your skeletal systems, your circulatory systems, your excretory reproductive systems, except for the germ cells, that's your egg and your sperm, and your primary oocyte and your primary spermatocytes are produced here. The dermis of the skin, under layer, not the outer, not your epidermis, which is your outer layer. And then the adrenal cortex, which is the internal part of the adrenal gland. Then you have your endoderm. This is going to be mostly digestive, okay? So all of your internal gut lining, your epithelial of your gut, liver, pancreas, all of the epithelial linings of your respiratory tract, your excretory tract, thymus, thyroid, parathyroid glands ultimately come from here. So think of it in layers. I mean, ectoderm is primarily your outer stuff. Mesoderm, sort of in between. And then your endo, your super inner. Okay? So there we go. So here's just a couple of things about some dividing lines of animals that are important. You got protozomes versus deuterozomes. So major divisions among this. Your deuterozomes develop in a way that's like your chordates and your echidnoderms. Okay, so a chordate is your internal skeleton like us. Protozomes is everything else. So pretty much everything else besides an echinoderm and us, and in invertebrate animals. And so differences is the direction of the way that cleavage happens when you form the gastrula, which is again that inner streaming, and the fate of the blastospore, which happens during gastrulation. Just bear in mind that development is, you could take an introductory development class for a whole semester in college, and I am giving you an overview in 20 minutes. So, bear in mind this is not the end-all be-all of development. I just want to give you guys, kind of like the background for it. So neurulation and organogenesis. So this is where we're going to start to form the development of the primitive notochord. I love that word. We're getting fancy now. Which is going to be your spinal cord and your main, nervous system. Obviously, this is in chordates. And then the formation of organs, which is your, organogenesis. So somites, right? Patches of cells giving rise to organs. What's so mean again? Body. Okay? So let's look at it from the standpoint of this. So neurulation in a frog embryo. Okay, what's really cool is as it comes around, all of those cells are going to stream around and they actually form this fold that's going to form. And then it zips. We even do the same thing. So we come around and we fold and then our spine zips right up the back and around. So you actually have that line that's going to come right around and form. Okay, and this is going to form with the formation of that neural plate. And you can see that right here in this picture. And go back and kind of, take a second, look through it. Okay, once that neural tube is actually formed, and this is one of the first things to happen, okay, then organogenesis is formed and you get somite formation. It starts. Check this out because this shows neurulation and the formation of that neural tube. So definitely go back and watch this. Star this, go back and see it. It's wicked cool. So organogenesis in a chicken embryo, it's kind of similar, all right? Again, these are going to form. You're going to form that neural tube, and then you've got the germ layers, okay, that are going to form the actual chicken, and then you get that super inner layer, which is going to help to form the yolk sac that's going to provide energy to the forming bird, okay? So there's late organogenesis where you can already start to see things that make sense. You can see the heart, you can see the eye, you can see the forebrain, you can see the neural tube, okay? Blood vessels, already moving blood through them. And why are they flowing out? I want you to think about that for a second. Why is the blood flowing out and then coming back? Why isn't it just staying within the chicken? Think about that. So morphogenesis is actually the cells changing their shape during development, okay? So again, that morph piece. So you're going to get things like the creating very differentiated, very specific type cells. Very important in developmental biology, okay? Again, that structure-function relationship. As they're being formed, these cells begin to change in order to initiate whatever function that they're going to deal with, right? So how you came to be. I know you were waiting for this part. Everybody wants to know, right? So early stages of human development. So let's take a quick peek at mammalian, right? So again, back to in the beginning, okay? You are that zygote, that first moment, right? So in mom, gamete comes out, you get the sperm. Now, fertilization actually occurs up in the oviduct, okay? For a little while, you're moving by yourself down to finally implanting inside mom's uterus, where then for the next, 40 weeks or so, you're being supplied with nutrients, oxygen, getting rid of wastes, all via mom. So by the time you actually implant, you're already at the blastula phase. You're a blastocyst when you embed. Okay, so you've already gone through all of that early stuff. So here's that breakdown. Once you reach the uterus, blastocyst gets there, you dive in, okay, because part of that is going to become the actual placenta, again, that barrier. You're not actually directly attached. Your blood, mom's blood, never mix. You get your nutrients, you get your oxygen, all via diffusion. You get rid of wastes, all of that stuff, all via diffusion. So it kind of sits like folds like this. Okay, so your placenta, mom, okay, you have a barrier. That's a protective barrier that will protect you against certain things. There are certain things that will diffuse across. Alcohol, if you smoke, certain chemicals will pass through. Other things won't. So certain things like viruses and things like that are protected against passing through. Okay, so once about 7 days after fertilization, you actually implant. So about a week, you're on your own. Okay, and after that week, boom, mom's going to give you a whole lot of help. Okay, once you're totally embedded, then you're going to form your germ layers. You're going to get an amnion that's going to attach in the formation of the placenta. Okay, so that implantation is really, really important. So as development progresses, at 5 weeks, you're about the size of a sesame seed. You look like an alien, not going to lie. At about 14 weeks in, you're about the size of a plum and wicked cute. Okay, and then at 20 weeks, you're about the size of a soda can. And at 20 weeks, that's halfway there. That's when you're halfway cooking. Okay, it remains really small. One of the things that's going to send mom into labor is the fact that you have run out of room and you have run out of energy. You require more energy than mom can sustain you, and you do a big chunk of growing during the end. Okay, the placenta plays a big piece in this. So you were born, but you were only the first birth. Then mom gave a second birth to the placenta because mom doesn't want to hold on to that. Okay, so it's a two-piece part here. You come out, then the placenta. It's a temporary organ. It's there to provide, again, that point where your blood, mom's blood, can get close enough for diffusion to happen, but never actually mix. Okay, no blood cells actually exchange. When labor actually happens, it's enormously hormone controlled. Okay, so you're getting estrogen from the ovaries that are going to start. Oxytocin is a big piece of this, and that's going to provide stimulation for contraction of the uterus, which is just a big muscle that's going to contract and contract and contract and whoop, push you out. Okay, and there's a whole series of positive feedback mechanisms that are going to help with this. Okay, so bringing it back, and then you're going to get negative feedback situations that'll happen after that. Okay, then you get, as it's going through, the placenta, you come out first, placenta comes out after. Okay, And again, that connection between placenta and uterus, everything comes through via the umbilical cord and right in through your belly button. Okay, so what goes where? So polarity and cell fate, how it's done. Major development questions. How do they know where they are in the embryo? How do they know what they should become? Okay. Roughly, cues from two main sources. Uneven distribution of protein molecules. Okay, we recognize this from the last podcast, right? Signals from nearby cells. This is the induction piece. So those are the two things that give us cues that are going to help us with that. Okay, so unequal distribution of proteins. This is shown in C. elegans, which is a tiny nematode worm. What's cool about C. elegans is that we've actually looked at, and you can follow, they have mapped the differentiation of all, whatever, 900 and something cells that actually form a C. elegans. That's not a C. elegans, just in case you were wondering. So you've got the fertilized egg, and you can actually track the differentiation of all of these cells, which is pretty cool. Okay, so there's that. Now, polarity in terms of a frog embryo is determined by cues present prior to fertilization and by the direction of fertilization. So what's going to determine up from down, anterior, posterior, right, left, all of that is determined by the poles of the embryo of those axes and where it happens. So take a second and look at that. And that where that first cleavage happens helps to distinguish that up from down, left from right. Okay? So what cells become what? The fate of these cells. Okay? In early stages of the embryo, we can actually stain them and find where do these cells go? What do they become? Okay? What's their fate? Are they going to be epidermal? Are they going to be a blastula? Are they going to be part of the yolk? Are they going to be, what's their job? Okay. So here's that map of the C. elegans. It's so cool. So you can actually track from that first zygote what actually becomes what in terms of cell differentiation, all to the very end and time after the actual hatching. And they're kind of cool because they are kind of see-through worms, and you can actually, they're cool. They wiggle and and they're kind of fun to look at. They're tiny. Okay. And then there's other experiments, funky frog fetuses, okay, manipulating frog embryos in early stages so that you disrupt polarity, and you can do it pretty easily, which is a little weird, so you can actually make frogs do weird things, okay? So if you mess around with these at various points in time and remove a particular region, okay, you might create an organism that lacks pieces, like a tail or like the belly piece or like whatever. You might, actually prevent those signals from happening. Transplantation of a region, okay, the dorsal lip onto another embryo can actually create duplication of what creates, and you actually get another embryo attached to it. That's a little weird too. Limb development. Chicken limbs development is dependent upon organizer regions. Okay, so you're going to get this signaling for an organizer region to then become the actual embryo. Transplantation of that region can actually lead to limb duplication. If you move one cell to another area, just like it shows over here in the where you can see the ZPA, that organizer region, you actually create a second limb. Wild, right? There it is. But what's true for one chordate may not be true for others. So here's the question. Are there any questions? Whew! Wild. So there's animal. Now we're going to move into plants. That's part 2. So stay tuned for part 2 and just focus on part 1 now, or move right into part 2 when you want to, or when it's ready. See you guys later.